Okay, good afternoon, everyone. Welcome to New York Tech Libraries Open Access Week. Today is Friday, October 24th, and this is our 12:30 p.m. Eastern Standard Time session. I say that because we are proud to welcome Dr. Gitanjali Yadav and members of the semanticClimate team. Dr. Yadav will be presenting her work with the evolution of with semanticClimate today. Geeta is a scientist at the New Delhi, the National Institute of Plant Genomes, and a professor of data science. And she is also a trustee of St. Edmund's College at University of Cambridge. Her work is on genomics and structural informatics with applications in climate conservation and food security. I am very glad to share this presentation today, and the recording will be made available afterwards. As a attendee, please share questions in the chat or the Q&A. I think we will have a good, lively session here. And please check back with semanticClimate frequently, as many of these tools are collaborative and meant to be shared for improved access to academic literature and parsing climate-ready reports. So without further ado, please join me in listening to Dr. Yadav. Thank you, Dr. Yadav.
Thank you. Thank you, Vanessa. Good morning to everybody on the other side of the Atlantic, and a good evening to all my colleagues in Europe and Asia. I especially take this opportunity to thank the New York Institute of Tech and Millie and Vanessa, above all, for inviting us, the team in semanticClimate, to speak at this session and provide an overview of semanticClimate. Should I just share my screen? Replace current?
Yes.
No, I would just like to
you
share
should
my
be able to share now. Yep.
Okay. Are you able to, oldest question on the planet, are you able to see my slides? So here we are today. And as many of us are aware, the world of research and academic exploration has undergone a remarkable transformation in recent years, thanks to the advent of open access, the idea of AI, FAIR principles, and above all, the indispensable role of libraries in supporting open access initiative. I hope to talk about my own research today and how open access has transformed our work, enabling us to bypass research barriers and even consider questions like who owns our knowledge. The Open Access Week actually is right now very timely, not just showcasing how libraries, they serve as crucial gateways to the wealth of knowledge, but also revolutionizing the way information is disseminated and accessed, playing a vital role in advancing scholarship, fostering innovation, promoting global collaboration, and so on. I have just returned from Brisbane in Australia, where data scientists from all over the world came together to meet at the International Data Week. And for me, it was my first visit to the Southern Hemisphere. And what I found most heartwarming was the way the Australian chief scientist opened that meeting with a respectful acknowledgement of the traditional custodians of the land on which we met. They paid respects to the elders, past and present, all Aboriginal and Islander peoples, honoring their enduring connection to knowledge, and more importantly, to the world's oldest continuous data system, which is telling a story. And so therefore, recognizing how ancient wisdom continues to guide, the ways we gather, share, and care for knowledge, I hope to talk about semantic today also as a story. And the story, as Peter and I always begin with, is how libraries have played a pivotal role in supporting the work we do and the open access initiatives. We like to begin our talks with an ode to S.R. Ranganathan, considered to be the father of library science and documentation and information science. And he was in India. We recently celebrated his centenary of having joined his first job at the University of Chennai. And what you see on the screen right now are his 5 laws of library science, revealing how libraries actively contribute to the expansion of open access resources, of organizing knowledge, collaborating, curating, meeting and connecting publishers and researchers for scholarly information, making such a wide array of research materials available to the academic community as well as to the society, public alike. So, my talk, the story about the work that we do, my talk is about, of course, about the theme of the Open Access Week, which is who owns the knowledge that we have, but also equally open access and the research that we do and the importance of open access in our research. So my work, our work, a large group in my team is here today. We work on the, phytochemical landscape, the dynamic mosaic of changing chemistry that surrounds us in space and time all the time. I was trained as a botanist, and I learned very early how plants have evolved some of the most advanced forms of communication on our planet. They communicate with each other, they communicate with the rest of the biosphere through chemicals. And unpacking these we call them chemical arsenals. It's not just about pushing and merging boundaries of modern technologies, but also about being able to understand and appreciate how the chemistry in the phytochemical landscape is pivotal to human well-being. Phytochemicals are all around you. All of life as we know it, as you know it, is complex chemistry in a semi-permeable bag. And our aim is to digitize, in semanticClimate particularly, is to digitize the entirety of recorded plant emissions using scholarly literature. This brings me back to the Open Access Week of the New York Tech Libraries, because it was on your first day of your opening this OA week that we were able to launch and announce the semantic encyclopedia. And that is where we wish to join hands and, have more joint workshops, trainathons, hackathons together. More recently, we have also combined our, joined hands, of course, with the Wider Earth Metabolome Initiative, an international mission to comprehensively profile the metabolome of all known species, so the chemistry of all known species on Earth, building a common electronic resource to share this knowledge globally. The position of the EMI team basically is at the crossroads of analytical chemistry, computational biology, ecology, and evolution, linking open science and open data, and placing us uniquely to generate a planetary scale metabolomics data resource. So our specific contribution to the EMI is a collation of a global repository of plant chemical fingerprints based on the data that we've extracted from over 13 million different published papers through an open science initiative that explores and establishes robust and scalable workflows for the digitization of all the world's chemo and biodiversity, from botanical collections to wild ecosystems and so on. And therefore, we believe that supporting the mission of the Earth Metabolome will greatly not just add to our own effort of deciphering the phytochemical landscape, which I talked about just now, but also give us the ability to employ artificial intelligence for identifying patterns in nature that build foundations for a framework that helps us understand and address climate change through these chemical networks that support life on Earth. And therefore, we hope to establish a global interactive living resource which fills the gaps in biodiversity and provides machine readable data for generations to come. So how do we do all this, and what do we do exactly? Some of you may have, if you've studied biology in your early years or are biologists or life scientists, you may have heard of the variable genomic code that all living cells are composed of. I would like to introduce you to the variable plant emission code. Okay. And so close your eyes and think about the scent of lavender in the unending fields of Provence. Or if you're in Asia, the familiar bluish tinge that you have in the air in the Himalayan foothills, or the yellow of sunflowers, or the flavor of cinnamon. All of these and much more that we cannot see, that we cannot fathom, are part of a variable chemical code of plants, and variability is the key word. So there are more than, several tens of thousands, more than 35, at least 35,000 recorded unique biochemicals that are produced by plants that are not required for the plant's growth and development. These chemicals, belonging to a chemical class called the isoprenoids, basically, they are released as volatiles, emissions, lightweight emissions, and they constitute the most diverse group of phytochemicals that we know and the one that we are investigating in the lab. Right? So billions of tons of volatile terpenes are being emitted annually by the plant kingdom on the planet, mediating so many different kinds of adaptations. They have many industrial uses also, such as biofuels, feedstock, fragrance, flavor, medicinal chemistry, but either way, coming right back to what we are doing about the variability in plant chemistry. It is not just in the structure of the emitted compounds, but also in their function, and is evident at all levels, as you can see in this image, within and between species, within and between individuals, within and between tissues, developmental stages, and so on. So basically, my talk here is today divided into two sections. The first part is about how we capture and measure the variability in emission profile. So how we get the data. We of course do it using a combination of real-time experiments and a lot of literature mining. So a lot of open access and library access is what we need. And the second part will be when I talk to you about what we do with all this variability. How do we study and analyze the variability that we find in the data that we record and observe? And what we do with the data that we collect. Essentially, that is where I talk about combining modern and traditional knowledge-based approaches. And that is how I hope to introduce our semantic toolkit and the semantic encyclopedia to you today. Okay, so first question, how and where do we get the emission data? Conventionally, we identify very small molecular weight compounds which are emitted as volatiles from a plant's headspace, either the flowers or the leaves or the entire plant, by performing a steam distillation followed by a gas chromatography and mass spec. For larger molecular weight compounds, we perform other experiments in the lab, and these processes have all been improved and improvised over several decades and several ways to capture the headspace of plants in the field itself, like live in the field rather than in the lab, by using SPMEs, portable GC machines like the Xenos, dynamic pumps, and so on. However, even under the most conducive conditions, there is no way that you or me or anyone else could experimentally analyze or measure the chemical fingerprint of each and every plant, species, and individual on Earth. But it is possible to do the analysis on a large enough scale so as to draw statistically significant patterns from the subpopulation that we study. And therefore, our premise is that much of the information that we seek already exists in published literature. So 15 years ago, we began this work. I met Peter 14 years ago. Was it 14 or was it 11? I think I've known him forever. But we began extracting and compiling plant emission data, me and my students, 15 years ago, in order to create the essential oil database. And the database basically is freely available to everybody on the planet. It's got more than 100,000 essential oil records with data spanning 100 years of published literature, representing 8,000 compounds and 2,000 species from about 100 plant taxonomic families. To give you a sort of idea of the kind of data format and structure that we use in this repository, it is not just the chemical compounds that are emitted by which plants. The question is, what are the compounds that were emitted in what amounts at the instance of collection from which plant species, but also contextual information about each of these emissions, such as the time of collection, the source of collection, the geographical location where that emission happened, the prevailing conditions, because the variability can be in any of these features, right? And what is the main shortfall that we have with this repository? The biggest shortfall is that it is merely representing a very tiny fraction of what we could potentially find in the full scholarly corpus of published data if only we had greater access. So that is why we started creating a gold standard, a series of. So the first strategy that we have is start extracting information from papers. So we've got a series, we've got hundreds, thousands actually papers, and we write a series of codes that is going to allow us to extract emission data from published articles directly without reading the paper. Now, that is another very important feature in the current day where you're expected to perform meta-analysis but not read papers because there are copyright issues, access issues, and whatnot. But mercifully, it is possible to extract knowledge without reading a paper, and that is where our semantic effort comes in. So this information, the one that we want, is usually available in tables inside papers. And these tables, more often than not, are in very, very different formats and diverse formats. Like, for example, here is a table from different tissues of lantana, an ornamental in the New World, but a very major aggressive invader in the Old World. So our most critical data is the name of the compound and the name of the plant, as you can see here. But we do need contextual information, I told you earlier, which is in the other columns of this table, sometimes in the other text of outside of this table, and so on. So there are different kinds of tables, as I can show you here. Unfortunately, the contextual information is so difficult to capture automatically. You need more comprehensive and more tricky code. And the plant species name is sometimes just not in the table itself. So you have to go back to the article metadata and different kinds of sources for this information. The other issue with collecting large volumes of chemical data basically involves complexities associated with data disambiguation and normalization. That needs a lot of careful attention in pre-processing. Here we are applying it to plants and the medicinal oils that they are making. So what we are convinced is that we'll be able to greatly increase the repository that we built with a search of the full scholarly corpus. And that is where we had some ray of light in this direction with the American technologist Karl Malamud, who gave us access to a gigantic store of article metadata extracted from 73 million journal articles, right? Dating from, guess how long back in the past? 1847. And up to the present day, of course. In order to avoid breaching copyright, we built code. That's where Peter and I began on Semantic 11 years ago. And we started building code that can crawl over the metadata, scanning through the world's scientific literature and pull out insights without actually reading the text. So all of this is very doable. And in order to build the code that would scrape literature, that's our second strategy, basically extracting knowledge from the complete corpus and about plants and their chemicals. For now, of course, we moved on to climate and much more eventually. So we then perform the scan, and this was done originally with the with the Florilegium data, and my student Preeti is now trying to expand it with the OpenAlex data. When Peter and I first started building the code to extract plant information, we were calling ourselves CEV Open. I'll show you a little timeline how we changed our names to eventually, evolve into semanticClimate that we are today. But from day one, we represent a virtual citizen science community, creating open and, more importantly, modular knowledge, which means the tools and the software and dictionaries that we create can read open published scientific literature and make it semantic. And therefore, what we do is to crawl the metadata for any plant species that we have and their parts. We have a full list of known plants and plant parts. We call these dictionaries. And I think earlier today, our program manager Renu explained to you the concept of dictionaries, for which I'm very happy to share the video. And I think, Vanessa, you would have the videos already about how we make use of dictionaries to help extract knowledge about a particular area of interest that we have. So we have dictionaries on plants, their plant parts, volatile compounds, locations, geography, anything else. The other issue that we have with collecting large volumes of data, such as chemistry, involves the complexities associated with disambiguation and normalization. Like I told you, how do you identify which are the medicinal oils and so on? So, you know, when we collect all of this information, the question comes in, what do you do with the analysis? And so here is a short snippet of what we are doing with the enormous amount of data that we collect and we have collected and we continue to collate. The focus of our lab is, for example, again, another little story, invasive species. One of the major side effects of the way people have been moving plants and animals around the world with the dawn of the liberal globalized world, that began originally in the Victorian era, of course, and has continued unendingly, is the introduction of invasive species or plant immigrants with no natural predators to control them on new soil. What you used to be calling a flower has become a weed for people in a new land. Some of the most aggressive invasives can be introduced into a very small area, and then they escape or explode out, competing for nutrients, for light, for pollinators, seed dispersers, crowding out other pre-existing species. And these, that's how species decline happens, species extinction happens. And a lot of climate change is also leading to some of the extinctions that we see around us today, not just in the plant world, but also in the animal world. Some of the most disruptive invasives have only become too familiar. So the Japanese knotweed and the rhododendron ponticum are very familiar in the UK, while invasives like lantana and ageratum, Prosopis juliflora, we call it the vilayati kikar in Hindi, are very well known in the Indian subcontinent and so on. So what we have done is to collate much of the information that we have pertaining to invasive species and we use that from the metadata that we did. And then we perform statistical pattern analysis on these. And here are some of the work done by our students. Abhishek and Preeti are the ones who have extracted the data, collected the ecological headspace. Renu processed much of this in the lab. Jamila added biotic and abiotic information, and Yuan built and tested the accuracy of machine learning classifiers. Mandeep handles the genomics, Kavita does the back-end coding, and a lot of interesting stuff that we were able to therefore map on the geographical emissions to the World Trade data and combine it with species identification records in the GBIF database, which is one of the largest repositories of species and their occurrence around the world. So we found a lot of support for the phytochemical expansion that we have by statistical comparisons of the emission profiles of ageratum between native and invaded habitats, as you can see on the entire map of the world. And this in turn provided evidence for one of the evolutionary hypotheses called the novel weapons hypotheses and so on. I also want to give one more story idea that we use a lot of pressed plant potential that we are interested in. I told you, there's a dazzling variation in plant chemistry, and that is the primary mediator of trophic interactions, herbivory, predation, parasitism, disease. At the same time, we also have to look at how these interactions feed back to influence spatio-temporal variation of chemistry in plants. And this kind of study can sometimes very nicely be done with botanic gardens. So it's both libraries and botanic gardens, I suppose, in my own way, that I want to say, that have been very useful for us in the work that we've done. So if you found these little stories interesting, please join us, because we are always looking for partners. You have to tell us what kind of topic you're interested in or your group is interested in, and we would very gladly, you could use our toolkit. It's a modular toolkit, as I said, and it can be used on any topic at all. And when I say this, the fact that it can be used on any topic at all, I'm using the example of climate change, because climate is not just a topic of science, life scientists, or legal topic. Of course it is, but above all, it's an issue of social justice also. And so what is it that we did together in order to, put our modular toolkit and make it something that could be used by anybody on the planet for accessing knowledge that you have in your corpus or scholarly corpus. And when I say access to knowledge, it is not just reading material, being able to, so what I'm probably trying to arrive at is that, is that if you look at just the IPCC report itself, the sixth assessment report, it's got 50 chapters, it's got 10,000 pages, and it's in what we call the locked format, or a portable document format, the PDF that was invented about 39, or was it 40 years ago, for people to be able to see data and figures and tables together in a human readable way. Today, the entire world is addicted, if not entirely dependent on the PDF format, so much so that we completely have forgotten that the PDF format is not something amenable to machine readability, or to machine access or analysis. And so therefore, if you have access to just the PDF, does that mean that it's available, or does that mean that it is accessible? You might say, oh, this is a bit of semantics, and it's probably no difference in the wordings, but it is a crucial difference, because accessibility is not the same as availability. Yes, you may have the library available to you, but is it truly accessible? Is the knowledge accessible to you? And what are we doing to convert available to accessible? And how are we doing it in our way? So basically, what we've been doing all this time is talking accessibility. I mean, what we've been talking is accessibility, but what we've been doing is merely availability. And therefore something needs to be done to fix this. And therefore, if you go back to the parental traditional original dictionaries, choose your favorite one. The definition of accessible is that it has to be, the information has got to be available, of course, machine actionable, exportable, comprehensible, and of course, where is that? Machine readable and machine actionable. PDF does not allow that. If any of you has ever used a PDF, the kind of layers of information that it has, ever tried copying and pasting from a PDF, you will find that it's nearly impossible to retain the format, nearly impossible to compare things. Overall, it's very, 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 very difficult. And therefore, just having availability of a PDF does not mean that we have accessibility to the information that we want access to. So what we have done within semanticClimate is to find a solution to this, and to achieve true social justice, or true justice for climate at least, through the fact that open access must encompass availability of the data to easily discover, download, and read it within an internet browser. Actionability, meaning having the expectation of machines taking their own analytical actions on the information that we feed to the machine. And exportability, so that the information can be transformed and combined with existing or other information from other sources and repurposed to generate new actionable knowledge. Finally, comprehensible is another definition that we have, so that we are, so we as humans can take action and understand what it is, whether we are researchers or policymakers, legislators, citizens, scientists, farmers, inventors, whoever we might be. So what we have done over the years is to open access to machines and to people. Peter and I first, at the turn of the, when the pandemic began, we started looking at the virus and studying the virus. We then moved on to, after the pandemic, we thought about studying and extracting, as I explained to you just now, plant information and emissions. In 2022, we began systematically addressing and semantifying the IPCC reports. And this year, thanks to you, the New York, the tech libraries, we've been able to, with your OA week, launch and announce the semantic encyclopedia for the first time. So that's where we are today. And what we've done is to convert data from not just machine readable, but also to machine ready through machine intelligence, through various kinds of transformers, machine learning concepts, and AI-assisted literature reviews. We have lots and lots of demos. We help people understand the back end and the front end of the way AI is used over natural language processing. And much of this work is converted into a hands-on, in your hands, on your laptop, a finger dippy kind of real, live hands-on work through the Google Colab that I'm sure you've seen earlier in the session that we conducted today. And through these little modular solutions, we make the data fair in the sense of findable, accessible, interoperable, and reusable, which was the original 2014 definition of fair. But the 2024 or 10-year new definition of fair, after 10 years of its first instance use, is that now we also want to make data FAIR, meaning fully AI ready. And that is the business that we hope to convey to each and every participant of this OA week, to each and every participant of the various workshops that we conduct. You have to make sure, think about your data. You've absolutely got to make it fully AI ready for it to be comprehensible, machine actionable, and readable in the current era going forwards, in the digitization era of 2025 plus plus. So overall, I believe through the toolkit that we have made, we have got a check mark or tick mark on each of the definitions of accessible that we did create. And I think I would like to just, I don't know how much time I have, but I want to give a small example of the fact that much of this work that we've done, the modular systematic toolkit that we've built, is on Google Colab. And the Google Colab notebook is such a brilliantly open e-notebook environment. You don't have any hassles with setup or versions. It's very human machine friendly. It supports interactive programming. It is very easy to learn and explore new tools within this one. And so therefore, we have this huge group and team of stars and clouds of students. We call them our young scientists in the age of knowledge neocolonization, where they are going to help us address open in its truest definition. And so Peter and I together have been creating Youth Knowledge Champions in semanticClimate. We welcome you to join this global community of volunteers, interns, mentors, partners, collaborators. We are very happy. We have a zero barrier to entry, and we are more than happy to have you become a part of us. The activities that we do within Semantic are basically 4 different activities, or in fact, I've put 5 here because of the huge collaborations that we are very keen to expand. But the 4 activities that we have are, of course, research. We perform AI over natural language processing. We are very keen on open science and knowledge liberation from different kinds of, I've given you examples from the phytochemical world today, but also climate access and climate change. Also before that, we were working with viruses. So any theme of interest that you have, we perform AI over NLP and knowledge extraction for that corpus. We also perform internships, which range from very short internships like 8 weeks going up to 12 months sometimes. And these internships are mostly about tech development, but we are very, very flexible in terms of interns who are not techies and who wish to basically help us in even gaining visibility for the work that we do. Then we have hackathons, of course. Our hacks are also very, we design them based on the group of people we work with. And these hacks are sometimes half day to 2 day long ones. In the 2 day hacks or even longer hacks, we usually have groups of teams asynchronously working alongside. This is what we've been doing in the Force 11 FISCI annual events, where Vanessa, you were there and you were part of this one. So you get to work asynchronously on use our toolkit and in some way to be able to extract knowledge or gain more sense about your corpus. And of course, we conduct a lot of events such as webinars, workshops, panels, discussion panels, and a lot of outreach, particularly in a country like India or the global south region of the world, where we have to cater to multilinguality, to rural students sometimes. It's been amazing how Peter has worked with rural students in India from small villages online, despite, the distances, the boundaries of nationalities, languages, cultures between us. I feel that somehow data and the human machine interaction allows us to shake hands across these crazy barriers that otherwise we would think are unsurmountable. Well, they are surmountable, and this happens only through collaborations like the ones we have with you this week, national, international, and regional collaborations. So the overall toolkit workflow, which of course our program manager Renu has explained to you already, is that we have these open access repos. We search them for terms that are of interest to us, let's say climate, and then we make a mini corpus, and through the mini corpus we generate dictionaries. Through the dictionaries we build on semantic literature, and on this we perform either learning or AI. And now, through the shifting clouds of young interns that we get, we do newer and newer things, such as the encyclopedia that Udita and Zainab have built this year. So here is our list of stars, very, very few, but just in the last couple of years, the people who have helped us. And the original, the first interns, Ayush and Shweta, who helped us begin the initial toolkit modular software tools. But now through the Jupyter and Colab system, or I call it a new term, we got a rig, an experimentation rig of the Colab and Jupyter ecosystem. We have now been able to perform AI-assisted literature review that our program manager Renu performs, as well as create the semantic encyclopedia that we've announced this year, right, this week, and so on. So here is an example of, a typical article on climate justice through the European PMC. We see a bigger picture using the dictionaries that we create. We enrich those dictionaries by embedding them into the global knowledge graph that is the wiki, and we bring in multilinguality for these. More than anything, what we do is learn by doing on this student authored toolkit. And what I see mostly from interacting with people and doing these workshops with people who we've never met before, also a lot of people come back to say, wow, this is easy, and this is so easy, and I've been able to wrap my head somehow for the first time around what's happening at the back end and the front end. And that moment of that Yahoo moment of understanding in somebody who hadn't understood till then what AI is all about or what these toolkits are all about is what we strive to constantly try and achieve. So we have the automatic indexing with our dictionaries, through which we have made every chapter of the IPCC semantic. And we earlier used to use an Obsidian-like graph that allowed us to be more interactive. But now you've seen the semantic encyclopedia that we've created. And overall, I would like to just say that is real open access, and that is real open science. And we would love to continue doing this with you and with like-minded people around the globe, gladly. So once again, thank you very much for inviting us as semanticClimate to present ourselves, our story, and our work to you, and especially in the form of a story. And I hope it made sense to some of you. We keep bypassing different kinds of barriers that I told you, and it all began when I first met Peter in 2014, and he's visited India more than once since then. We'd love for you to meet Amy, our mascot, and you can find out more about us on LinkedIn, on our website, on Google, on GitHub, and so on. Thank you. Thank you very much. I'm going to
Thank
stop.
you. Thank you, Geeta. That was incredibly generous and fantastic presentation.
I hope it makes
I'm
sense.
just so impressed with all of the methods that we could apply any of these tools and really wrap our head around how we even synthesize information, like you said, in
Yeah.
our own corpus. We have a question in the chat, if I can read it out loud. Naomi says, I need to start with a big kudos to you and your team. This is really impressive. I have a knowledge on these topics, so I want to know what all of the limitations of the GC-MS that you all faced with deep learning. And I'm sorry, I don't know what GC-MS means.
Oh, my bad. I'm sorry. That was a very specific term called for, botanical and chemical in the world of chemistry and botany. It's called gas chromatography, followed by mass spectrophotometry. And these are terms. So we've had limitations because we don't have access to all the species and all the parts. For example, Nayanu, we are very keen on understanding invasive species, as I told you. And we are, for example, lantana is one of the most aggressive invaders. Now, in Delhi, where we work and we live, there is one particular subspecies, or I would say one particular variety of lantana, but India has hundreds of these cultivars of lantana, and each one is releasing a different bouquet of chemicals. Now, it is not possible for us to physically go and find and identify and then extract the chemistry of each and every cultivar. However, different people living in different places have done this and recorded this in publications, and therefore we use libraries and open access, and that's how we overcome the limitations of the human limitations of not being able to do every single experiment through meta-analysis of existing published records. Does that answer your question? I hope so.
Diana, I think you had your hand raised for a moment. Did you want to chime in? Did you have something else you wanted to add? Okay.
Oh,
No.
I see a question. I see a question by Millie.
Oh, great, great, great.
Yeah, the link to the invasive species project. So we've been working on the on lantana camera in the lab, and we usually publish papers. So the thing is, we don't I wish we would create links like that, but what we have is we have published different papers. We are in the process of writing some more. So we've looked at lantana above ground and below ground. Above ground, we've looked at it in the Himalayan ranges, as well as in the urban regions in India, as well as in the Andaman Nicobar Islands. And below ground, we are trying to address the question of does the soil where lantana grows, is that soil somehow modified to allow lantana to dominate that region or that habitat? And what we are finding, Millie, is incredible that when you do the genomics of the soils, you find the microbes that are there. For example, you might have heard about the gut microbiome and the human buccal microbiome and so on. But even in the soil, even around the leaf, there are these little microbiomes where zillions and gazillions of bacteria and fungi live, and they make these networks in the soil. And we are trying to see whether, and lantana or invasive species are making use of these underground networks, fungal networks and bacterial networks. And we are trying to decipher if these networks are somehow making the invasive species more invasive or helping them in their success to dominate a new habitat. Does that answer your question? Sorry, that was too much information, I suppose. Okay, okay. Yeah,
That's
Peter.
great. And yeah, yes, it must have been very time consuming.
Research is like that, and we are more than happy to. So the work we do with semantic is essentially the whole idea is essentially to help our own researchers to be able to use the modular toolkit to answer questions easier. But of course, as I said to you, the 4 parts of what we do in semantic outreach, as well as events, as well as internships, have now become a part and parcel of everything that and therefore we need your help, and we need your to collaborate with you and others like you, or like-minded people who can help us expand the work we are doing and get others to do their research better, like we've been able to do our research better with the same toolkits.
Fantastic. Fantastic. So I wanted to share some of the recording from earlier today. If there's not any other questions, I'll start to queue that up. And Peter, if you wanted to add anything, you can unmute yourself and do that.
Yes, please, Peter.
So just to say what I put in the chat, that I'm really excited to have this opportunity, because this, of course, is something that we can showcase to the world. And I think libraries have a really central role here. I've been talking yesterday at length to our open research team in the Cambridge Library. It's fairly early stages, but what has happened here in this week is something that can give them and us a chance to develop new ways of doing things. So I'm thinking of hackathons. And one thing I would recommend to think about is the idea of having librarians embedded in scientific departments, because that's a way of spreading knowledge and both sides getting up to speed with ideas and technology from the others.
Yeah, I agree.
Yeah, Peter, I would say that, I'm an architecture librarian, but I think the methods of synthesizing information are just, they continue to change, and with depth of knowledge that you can't really stay within your subject area and expect to be able to understand all these different processes. So I'm going to, for this portion of the webinars, I'm going to share a portion of the presentation from earlier. And if the panelists want me to advance to any particular section, please let me know. But I am going to just start from, going to switch to full screen, just make sure you can all see that. And I'm going to share this presentation. Thank you.
I can't hear any sound, Vanessa. Is there a sound with this, or is this just a series of slides?
Let me adjust that. I tested it this morning and we had sound, so let's see. Make sure. Original sound for musicians. Let's fix, we don't want. Okay. Okay, I'm just going to try again. It had been working. Oops, I'm out of here. Oop, how many pounds? Okay. Let me pause. Anything else? I think that we can, like I said, I can distribute all this quickly, and then we can conclude here, and and be able to continue to disseminate this information and talk again next week. I really want to thank everyone again for joining today. It was really excellent to share this, to share at this stage with semanticClimate. It's such a huge system of informatics that it's so important to bring this into so many different contexts. So I want to thank everyone. Oh, let's see, we have one more thing in the chat. What do we have here? Takes about 3 months for non-CS students to become productive with their toolkit. Okay. And add to it. Excellent. Okay, everyone. So I want to thank you all again. Dr. Yadav, Dr. Peter Murray Rust, everyone who was able to join today.
Thank
I
you.
wish everyone a great evening and afternoon.
Good night from our
Good
side.
night.
It's all
Yes.
in India.
Oh
Thank
my
you.
gosh.
Thank you again. Okay.
All
Thank you,
right.
Amy. Bye, Amy.
Oh, bye, Amy. Thank you, everyone. Okay. Bye-bye now.